Alright guys, this is the second part for your Unit 3 test review. We're going to look at Chapter 8 and Chapter 9, which is asexual reproduction is Chapter 8 and sexual reproduction is Chapter 9. Remember that asexual reproduction is with one parent creating genetically identical cells, whereas in Chapter 9 with sexual reproduction, it's where we're going to take two, where we're going to have a mixture of a egg and a sperm in order to create the new individual, which gives variation and uniqueness. So as we take a look at this, guys, there's a couple of terms that you should know. Diploid and haploid are these two terms. Diploid, guys, is a full set of chromosomes. This is represented by 2N, and the reason it's 2N is because it can be different for every organism. That N is a different number for every organism, so we're going to look at that in a second. Haploid means half a set of chromosomes, so you only have N that's present. So if we look at this over here in this chart, you'll see that we have organisms listed. You have the body cells, which is the 2N, that's the diploid, versus the gamete. Guys, remember gametes are sex cells, whether they're egg or sperm. And so when we're looking at that, it's going to have N, half the number of chromosomes. So guys, be able to calculate this. So if I gave you something like this and I said the human has 46 chromosomes in their body cells, that's the 2N. And I asked you what were the number of gametes, you would take 46 divided by 2, which gives you 23. So you should be able to figure that out. Now on the other hand, if I give you like the example with the garden pea, if I give you the gamete, which is N, you should be able to find 2N by doing 7 times 2, which is 14. Okay? So with the fruit fly, if I give you the 2N, you would do 8 divided by 2, which gives you the gametes of 4. So the haploid is 4. In a tomato, the diploid is 24. Therefore, the haploid would be 12. In a dog, the haploid is 39, so the diploid, you would have to do 39 times 2 would be 78. In a chimpanzee, the diploid is 48, which means the gametes, or the haploid, would be 24. In a leopard frog, we see that the gamete is 13, which means the body cell would be 26. And in corn, the gamete, or the haploid, is 10, so the diploid would be 20. Okay, so make sure that if I give you an example, you should be able to either divide by 2 if you need the haploid number, or multiply by 2 if you need the diploid number. So this is just giving you an idea of how to calculate that. Now, another set of terms that you need to make sure you know are the different types of DNA terms, whether it's chromatin, chromosome, or chromatids. So when we look at this, guys, a thin strand of DNA is chromatin. It's thin, it's invisible underneath the microscope, it's the thin form of DNA. Chromosomes, on the other hand, are condensed. They're coiled around histone proteins, and so they're more visible. So it's condensed, it's darker. Now, chromatids are replicated chromosomes. A lot of times we call these sister chromatids, where there's 2 of them together, as you see here, and they're held together in a center area called the centromere. Guys, centro means center, mere means part. So it's important that you be able to identify this. Whether we're talking about the chromosome, that's called a sister chromatid, or the middle part, which is the centromere. Okay, so this is some more of those definitions for chromatin, chromosome, and chromatids. Alright, this brings us to the cell cycle. Now guys, in the cell cycle, you need to understand the different parts, and you also need to know the checkpoints that are kind of seen in each of these to allow the cell to continue through the cycle. So guys, the G1, make sure that you know what's happening in this G1 stage. This is going to be where organelles are replicating and the cell is growing in size. The S phase, which you can see here that's kind of in the green, this is where the DNA gets replicated or synthesized. We're going to make new DNA for the new cell we're going to make. We also see there's a G2 stage. The G2 stage is a protein synthesis, final growth preparation so the cell can get ready to divide. Now it's important to note that there are checkpoints at the G1 and the G2 phase. At both times you're going to check to make sure that things are correct and moving smoothly before the cell can continue on its division process. G1, S, and G2 are all part of what we call interphase. Okay, so they're all part of what we call interphase, which is really important to note that this is not part of mitosis. This is just part of the cell cycle. And it's the longest. This is where the cell spends most of its time. The other part is mitosis. Mitosis is what we call the M phase, and this is where the nucleus is going to divide. It's the nuclear division. Now, this is represented by a couple of phases that we call PMAT. If you can remember that it's PMAT, that's the correct order. Prophase, metaphase, anaphase, telophase. So you have those four, and they're called PMAT together. Now, the last thing that happens is cytokinesis. This is where the cell has the two nuclei now, but it actually divides. It pinches off into the two cells. This is where the cytoplasm is going to be divided. That's cytokinesis. There's also a checkpoint here in the M phase that happens at metaphase, making sure that those spindle fibers are attached to the chromosomes before it moves on. Okay, so it's important to know what happens in each phase of the cell cycle, and also the checkpoints happening in interphase at G1 and G2, and the checkpoint that happens in mitosis at metaphase. So let's take a look at what happens in each step of mitosis. Now, you looked at this in lab and you actually filled out a sheet where you were supposed to draw the pictures of what's happening, as well as give a brief description. This is a good study tool, guys, so that you can be able to identify them either based on a picture or a description. So I'm going to put some pictures up here, and then I'm also going to describe what's happening in each one. So the first stage of mitosis is prophase. Well, here in prophase, a number of things are happening. Okay, the first is the chromatin is starting to condense into chromosomes. So we can actually see them in that X-type structure. The nucleolus is disappearing. That area inside the nucleus that's going to make ribosomes, it's going to disappear, lighten up. We're not going to see it, okay? Also, the nuclear envelope is fragmenting. It's breaking apart. You can see that here in the picture because it's represented by a dotted line. Also, the centrioles are going to migrate to opposite poles, okay? The opposite sides of the cell. Okay, they're going to migrate there. And the spindle fibers are going to start to form. And you can see that here that they're connecting those two centromeres. Okay, or those two centrioles. They're connecting the two centrioles. Now, I also have here some pictures. You can see that an animal cell is going through prophase as well as a plant cell going through prophase. All right, so this is showing you prophase. The second step is metaphase. With metaphase, the main thing that's happening is the chromosomes are lining up at the middle, the equator. Okay, the middle part of the cell. This is also where the spindle fibers are going to start attaching to those chromosomes. All right, so they're lining up there in the middle. Again, I have a picture that shows you the metaphase and in animals and in plants. Our next phase here is anaphase. Anaphase is where the chromosomes are actually going to start being pulled apart. Those replicated sister chromatids are going to migrate to opposite ends of the cell. They're pulling it apart, moving to opposite poles. Again, you can see this in animals as well as in plants. Telophase is where we've actually taken those chromosomes and moved them to the opposite sides. So those chromosomes are starting to unwind back into chromatin. We also see that the nuclear envelope is reforming. The nucleolus will reappear. The spindle fibers begin to disappear. And the centrioles have done their job now, and they're going to kind of disappear, not be seen as much anymore. Right? And so again, you can see this in animals, and you can see this again in plants. Now, we haven't fully divided the cell, the cytoplasm, because that takes place in cytokinesis, but we have separated all of the chromosomes by this point in telophase. Now, I told you in class that there was a way to kind of look at this as we were going through the process with your hands. So we have here prophase. Prophase, we have the big cell. It's getting ready to divide. We can see the chromosomes. They're your fingers. In metaphase, they line up in the middle. Okay, they're lined up in the middle. Anaphase, they're pulling apart. And then telophase, we start to form our two complete cells along with cytokinesis. Now I have two cells that are smaller than my original. Okay, so this is just a form of hand motions to help you remember those phases. Now, talking about cytokinesis, it's really important to know the difference between what happens in an animal cell versus what happens in a plant cell. Now remember, in an animal cell, we're going to see that they're going to pinch together like we see here. It's going to create a cleavage furrow so that they can pinch apart and pull apart. Okay, so it's important to know that that's a cleavage furrow. Plant cells can't do this. Okay, they can't do this because of the structure of the cell wall. They can't just pinch apart into two. So instead of having a cleavage furrow, we're seeing that they're going to form a cell plate. They're going to build a cell plate, which is then going to separate those two new cells. All right, the next thing we're going to look at, which is going to actually finish up Chapter 8, the main components of Chapter 8, is we need to be able to tell if it's a cancer cell, which is also known as an abnormal cell, it's not normal, and a normal cell. Okay, and this is going to be a type of matching. So when we look at this, is the cell differentiated? Differentiated means that it's doing its job. Okay, in a normal cell, yes, it's doing this. However, in a cancer cell, it's not functional. It's not doing its job, whether it was a liver cell, a heart cell, a lung cell. It's not doing that job anymore. Okay, the next thing we can look at is the nuclei. Most cells are supposed to have a single nucleus. So in a normal cell, they're uninucleated. However, in an abnormal cell, they become multinucleated. They have tons of nuclei kind of scattered in there because they're bypassing all those checkpoints we talked about, and they're just dividing uncontrollably. The next thing is apoptosis. Remember, in apoptosis, that's programmed cell death or cell suicide, where when the cell first gets messed up, if it's a normal cell, it will remove itself. It will get rid of itself. So normal cells do apoptosis. However, cancer cells, they don't. They don't care that they're messed up, and they're just going to keep going through the process of dividing. This brings us to contact inhibition. In contact inhibition, normal cells, if they have contact on all sides, they stop dividing. So they have contact inhibition. But cancer cells do not. Since they don't care if they're being touched on all sides, they just keep dividing, and this is what causes it to be a mass or a tumor. Okay, because it's just growing and pushing on all the other tissues. That brings us to growth pattern. In normal cells, they grow in organized single layers. However, in abnormal or cancer cells, they are disorganized in multiple layers, and this is again what creates a mass. So this is going to be one of those that's a matching. So on the matching, if I was to say this type of cell undergoes apoptosis, you would pick normal cells. Or if I said the growth patterns for this cell is disorganized, that would be cancer cells. Okay, so you're going to use this as a more of a matching. All right, so now we're going to switch gears and we're going to move into sexual reproduction. Now you're going to notice that this has some big similarities to what we talked about with mitosis. However, we're going to focus in on the meiosis. Now meiosis has two steps. It's got a meiosis 1 and a meiosis 2. The whole point of doing two divisions is that it's going to go from a diploid organism, 2N, a diploid cell, to haploid. This is going to create gametes, which are egg or sperm. Remember though, if we're going to create egg, it's made through oogenesis, and if we're going to create sperm, it's through spermatogenesis. So the purpose of meiosis is to produce gametes and half that number of chromosomes. On the other hand, another process that takes place in sexual reproduction is fertilization. Fertilization is going to produce a zygote. When we look at this zygote, this is going to be where we fuse together an egg and a sperm, and this is going to create a new 2N organism. This new 2N organism is called the zygote. This is the first cell after an egg and sperm fuse together. Now, along the same lines of terminology, we need to look at the process of crossing over, because this is something that's unique in meiosis. So guys, crossing over is when those homologues, remember homologues are similar chromosomes. So you've got one from mom, one from dad. They're similar chromosomes. They come in contact with each other and they do what we call the synapsis. Since they come together in a synapsis, which you can see here, they're going to exchange some of their genetic material. Now, it's important to note that this is only going to happen during prophase 1 of meiosis 1. That's the only time this is going to happen. So if I ask you a question on the exam that says when does crossing over take place, you need to pick prophase 1, because that's going to happen in meiosis 1. It's the only time crossing over takes place. This whole idea of crossing over is because of this structure called the tetrads. Guys, tetrads are only found during meiosis 1 of prophase 1 and metaphase 1. That's a really distinguishing factor so that you can tell that this is a step in meiosis 1. So this brings us to looking at those steps in meiosis. So let's look at meiosis 1 first. So you have your prophase 1. In prophase 1, we're going to see that the homologous chromosomes are going to pair up. Okay, they're going to come in contact and create those tetrads we talked about. We're going to see the same stuff happening, though, besides that we saw in mitosis. The nuclear envelope is going to disappear. The spindle fibers are going to form. The centrioles are going to move to opposite ends. The nucleolus disappears. So we're going to see all those same things, but the unique thing here are those tetrads, those homologous chromosomes pairing up. Then you notice here in metaphase. In metaphase, we're going to see that these homologous chromosomes, these tetrads, are going to line up in the middle. So metaphase still is lining up in the middle. But instead of having just one X in the middle, you have two X's next to each other in the middle. Now those X's are going to be pulled apart. Okay, one X going one direction, one X going the other direction in anaphase 1. So in anaphase 1, those homologous chromosomes are going to separate. Okay, so in anaphase they're pulling apart, but we're going to separate those X's. Again, in telophase, we're going to see that the nucleus reforms. The nucleolus is going to reappear. The chromosomes are going to unwind into chromatin. We're going to see that the spindle fibers disappear. Cytokinesis is going to take place, and now I have two cells. This is going to lead us into a stage called interkinesis. It's not interphase again, it's interkinesis. So in this stage, there's no replication of the DNA. It's just going to tie us from meiosis 1 to meiosis 2. So there's no replication of the DNA here. So then in meiosis 2, we have two cells now going through the same process. Both of these cells are going to do prophase 2, where the nucleus is going to disappear. The nucleolus is going to disappear. The centrioles are going to move to the opposite ends. The spindle fibers are going to form, and the chromatin are going to condense into chromosomes. Then we go to metaphase 2. In metaphase 2, the chromosomes are going to line up in the middle, just like we saw in mitosis. They're going to line up in the middle, and they're going to get ready to connect and pull those X's apart. Okay, so not the whole X going one direction. We're going to pull them apart where one side's going to go one direction, the other's going to go the other direction. In anaphase 2, they're pulling apart. Okay, so the chromosomes are pulling apart like we saw in mitosis. And then in telophase 2, we're going to see that cytokinesis is going to start happening, as well as reforming the nucleus. We're going to have the nucleolus reappear, and the spindle fibers are going to disappear. One thing to note, though, is that there are two cells again going through this, which is going to create 4 cells by the end. So guys, one way to actually tell if you're going to be doing mitosis, meiosis 1 or meiosis 2 is look at what the number of cells going through the process. If there's 2 cells going through the process, you're looking at meiosis 2. And meiosis 2, guys, is most similar to mitosis. So let me repeat that. Meiosis 2, when you look at it, is very similar to mitosis. There's just 2 cells doing it. So we've half the number of chromosomes now. All right, so this is going to create egg in oogenesis, and it's going to create sperm in spermatogenesis. Now what we want to look at is genetic variation. Okay, there are 3 ways that we get genetic variation through sexual reproduction. The first way we've already discussed, and that's crossing over, where we're going to see a different combination of genes because they have switched pieces. So that's the first thing. The second is going to be what we call independent assortment. Now, independent means that they do it on their own. They're not dependent on anybody. And assortment means we're going to separate. So when we look at this independent assortment, tetrads are going to line up however they want. Okay? All the dad ones are not going to line up on one side and the moms on the other. They're going to be mixed up. They can line up independently. Okay? And so that's going to be very unique because you may have a dad chromosome, dad chromosome, dad, mom. Okay? So they can line up in different ways. Okay? And it's going to be different every time they do the process of meiosis. And last is fertilization. Fertilization is a random fusion of an egg and a sperm. And since this is going to be random, it's going to be unique. Okay, so you have 3 things that happen here for variation. Crossing over, independent assortment, and fertilization. Now, the next thing that's important to look at is a mitosis versus meiosis kind of structure. What's happening here? Because this is going to be like a matching, and it's going to say, is this mitosis or is this meiosis? Now guys, mitosis is asexual reproduction. It produces daughter cells who are genetically identical to the parent. Now, the reason this is a good thing is it leads to genetic stability. So we want it all to look the same if it's like within your own body. So this is going to occur in what we call stem cells or body cells. And there's one division, so they divide one time, creating 2 cells. On the other hand, if you look at meiosis, meiosis is going to be sexual reproduction. It produces haploid cells. So we're going to go from a 2N down to N. This is going to mean that they're going to be each unique, which leads to genetic variability. This is going to happen in your germ cells, the gonads, whether it's testes or ovaries. We're going to see that there's 2 cell divisions. So since we're dividing twice, we're going to create 4 cells instead of 2. All right, so this is going to be one of those matching again that says in this process, there are 2 nuclear divisions. Well, that would be meiosis. It might say this process leads to genetic stability. That would be mitosis. This process makes 2 daughter cells. That's mitosis. So you can see how this is going to be used on your exam. Now, the last slide I have for you is the diplonic life cycle of animals. Okay, so you need to be able to look at a picture kind of like this and be able to label what's happening. Okay, so if we look over here, if we have the male and we're going to make the sperm, we're going to make the gamete, this is called spermatogenesis, and it's a type of meiosis. If we look here at the female, we're going to see that it's going to be a type of meiosis called oogenesis, which is going to create the egg. So now I have sperm and I have egg, and when they fuse together, that process is called fertilization. This fertilization restores the chromosome number back to 2N. This first cell that's created is called a zygote. Now that zygote is going to go from 1 cell to 2, that's through mitosis. It's going to go 2 to 4, that's also mitosis. But then you're not made of just 4 cells. That's going to continue to divide to create you as that new organism, whether you are male or female, starting the process back over again. Okay, so you should be able to label spermatogenesis and oogenesis, which are types of meiosis. You should be able to label fertilization. You should be able to label the zygote. And you should be able to label where mitosis is happening in this cycle. All right, so this covers pretty much chapter 8 and chapter 9 as well. Guys, please pay close attention to your drawings that you did in lab. Okay, we don't do that for busy work. That's to help you study. Not only are you drawing the pictures, but make sure that you're also writing a brief description that's happening in each one. It's to help you study for your exam. Again, if you have any questions or concerns, please feel free to contact me.